Hi, my name is Ben Newman, and I am a coronavirus researcher. And I'm going to try to use that to answer your questions. All right, so our next question is from Joy. Good morning, Joy. Let's see, is there any idea how long immunity lasts after getting COVID? Yeah, okay, fine, we can look at that. How long it lasts after getting the vaccine? We can start to look at that. A question answered yesterday said it varies from person to person. Totally the correct answer, but a very unsatisfying answer, isn't it? Yeah, just like, yeah. But it's the truth. Some people get the virus or get vaccinated and end up with no detectable immune response pretty much right after they have had it. It is a mystery how they managed to clear it or how their immune system managed to miss the big glowing red beacon of a vaccine that was there. But yeah, stuff happens. Human body's a weird place. All kinds of little nooks and crannies. um, yeah. How long does immunity last? Well, okay. So the older data. So we're not far enough in that we can really say for sure. So if immunity only lasted a week or something, I guess we would know by now. We know that in the short term, we've had people that got reinfected with SARS coronavirus something like 3 or 4 months after their initial infection. And this was lab confirmed in both cases. And they appeared to be better in the middle and it tested negative. So it was probably two separate infections. And there are enough of these cases where the virus was actually sequenced the first and second time. And we can be sure that it was not just the same virus that stayed there, kind of ducked its head down, then popped back up. Yeah. Um, yeah, so, yeah. Um, so that's like the worst case scenario. What's the average scenario? What's the best case scenario? We don't know for sure. For SARS coronavirus 1, though, it was found that you could detect antibodies in people who had actually had this. And remember, there were only like 8,000 people that ever got the virus, and 800 or so of those died from the virus. So, yeah, 7,200 survivors is not a lot of data. It's a smaller number than in any of the major vaccine phase 3 trials, to put that into perspective. But it was found in a sampling of those that you could detect antibodies for somewhere between 2 and 3 years after infection. And so, based on this, we can say that, okay, it could be a reasonable estimate would be maybe you would have some marker of immunity for 2 or 3 years. Question is it enough immunity to actually protect you from infection? And the answer to that is we don't know for sure. And if you think about it, what would the experiment look like that would actually prove that? So, if you had people and you had different levels of antibody in them, you could then, yeah, deliberately expose them to virus and see who got sick. But that would be a totally unethical experiment to do. And if you tried to do a similar experiment like that in mice, you could do it, but mice are not people, and there aren't any of the animal models that is a really good exact match for what the SARS coronavirus 2 does in a person. And even then, there is such a range for what it can do inside of a person that, yeah, I don't even know that you can match that with any one model. You'd probably have to have 20 different models for everything from asymptomatic to super severe, and sort of involvement of different organs and different parts of the immune system. And then you fundamentally run into the problem that a mouse is not exactly a human. It doesn't work in exactly the same way. They have some different parts of their immune system. Some of the toll-like receptors. Yeah, I don't know why you would care about toll-like receptors, but yeah, there's a couple of those with definite differences. As an example, there are different kinds of invariant T cells, which can potentially be helpful in an infection like this, that would be different. You just have different availability and different types, in mice and people. So, yeah, there's no way to really be super certain. If we are guesstimate, and I think the last part of the question does say if we had to guesstimate, yeah. So what we know is that from the studies that we've seen, so you have when you get about 20 neutralization units, and this is a sciencey term for a particular amount of virus that works to a certain extent in a particular kind of test in the lab. We could go into it, but I don't know that you need to. About 20 neutralization units above background is where the average person is at the time, 2 weeks after vaccination, when we think that protection seems to kick in. So that's a reasonable correlate. We can say, okay, if we're 20 units above baseline, that's probably going to be roughly for the average person almost, yeah, where you would be protected. And then you can see how much antibody a person makes, like the total. Because we don't just go to 20 and then stop. We go to, 500 or something like that. And then you can see in other studies, okay, once we have antibodies and over 3 months, how much did the antibody levels drop? Because they always drop, but, they drop at a certain rate. So they drop to maybe half that level in young people to maybe a quarter of that level in very old people. And so you've got a range there. You say, okay, for younger people, it's going to last longer, and you would probably be able to count on something like a year of protection for the younger people that are vaccinated and did get protection. Because remember, not everybody gets protected by the vaccine. Yeah, the vaccine does its best, but sometimes your immune system just won't listen. For older people, you're talking about something like maybe 4 to 6 months of pretty good guaranteed protection on average. I guess it's not guaranteed. Nothing's guaranteed. But this guesstimate that I'm giving you is probably a little bit pessimistic because it's only based on a certain subset, a little tiny proportion of antiviral antibodies called neutralizing antibodies. And real immunity involves a whole bunch of other types of cells, some of which have memory versions that last for a long time. But there are other situations, like in the flu occasionally, where these memory versions will burrow into tissues and hang out there, and they'll end up in the wrong place. They end up being very hard to call back after they've been made and sort of set aside. And so, yeah, this is essentially a question that can only be answered in the fullness of time and within the real world. We'll basically have an answer when we get to the point where the vaccine is no longer effective, if the virus is still around. And I hope it's not. If we can vaccinate everybody within this window, and I guess we don't know how long the window is, but the window is going to be somewhere between sort of the shortest amount of protection, which might be like 4 to 6 months, and the longest amount of protection we can count on, which is probably just a little over a year. So, yeah, 6 months to a year maybe is our window. And if we can vaccinate just about everybody in that time, the virus will go away, won't have anywhere to go. And so we'll never find out the answer to this question. And I think that is actually going to be the best answer of all. So if we can drive this dumb virus to extinction, then, yeah, that's something that we can look at in the lab at our leisure in later years and try to figure out. And I don't know that there will ever be a satisfactory answer unless we really mess up the vaccine rollout and the response. And I'm hoping we don't do that, and I think we won't do that because I don't know. Maybe I'm an optimist, but I think optimism is warranted in this case. There. So that's 50 cents worth of answer for I don't know. Yeah, maybe 2 cents worth of question. Yeah, I think that's more than what you wanted, but not that it's more valuable, just that it's a lot. So I'm hoping that helped anyway, or answered your original questions and some of the follow-up questions after that. Anyway, for now, thanks very much. Good question, and this has been Ask Dr. Ben.